So this is a TRP operon. So there's the promoter, the operator, and the genes of the operon. So since the operator is by default on, RNA polymerase can transcribe mRNA. This mRNA that's been transcribed will have the genes of the operons, the TRPE, TRPD, TRPC, TRPB, and TRPA. From here, the mRNA is then translated to polypeptide E, D, C, B, and A. These polypeptides are then bound together to create the 3 enzymes that create tryptophan. Simultaneously, while this is occurring, a promoter and a regulatory gene upstream transcribes mRNA, which is then translated to an inactive repressor. As this is going, tryptophan is starting to build up. And when it builds up to a certain point, then it starts binding with the inactive repressor, making it an active repressor. From there, the active repressor then binds to the operator, which causes the ceasing of tryptophan being created. Start again. Okay, so this is a operon when lactose is inactive and not present. So this regulatory gene then transcribes mRNA, which then translates to an active repressor. That active repressor then binds to the operator, which causes RNA polymerase not to be able to transcribe anything. In this case, there's lactose present. So when lactose is present, so in this situation, there's lactose present. So that causes lactose in the isomer form of allolactase to bind with the active repressor, causing it to become inactive. Since that happens, the RNA polymerase then binds and is able to transcribe this new RNA from this DNA. There's the genes that it codes with are LACZ, LACY, and LACA. These code for B-galactosidase, permease, and transacetylase. The first one, B-galactosidase, hydrolyzes lactose to galactase and glucose. The second one, permease, codes for permease, the enzyme that allows pretty much lactose to enter the cell. This one, the final one, transacetylase, really has no known function in this whole system. Here's a operon in the E. coli cell. So when there's a high amount of glucose and a low amount of CAMP and lactose, the CAMP is inactive. Since it's inactive, it's not able to bind to the operator and the promoter. And so, because of this, RNA polymerase can only transcribe very small amounts of mRNA. In the inverse scenario, where there's a high amount of lactose and a high amount of CAMP, but a low amount of glucose, CAMP is able to bind to inactive CAMP, making it become active. Then it binds here, and then RNA polymerase is able to transcribe new RNA pieces. When lactose is in the system, it doesn't affect anything due to the fact that it binds to the inactive lac repressor. Okay, there is the pre-mRNA. It contains a 5' cap, 2 exons in this example, and an intron. So the 5' cap stops the pre-mRNA from degrading. The 2 exons are the part of the gene that's expressed, and the intron is the part that's not expressed. So in order for the intron to be taken out, it has to enter a spliceosome. And it's helped by snRNPs, which contain proteins and snRNA. That plus other proteins enter the spliceosome and cause, at a intron-exon junction to cause the intron to be taken out. When it leaves this spliceosome, the mRNA is, with its two exons are bound together, with it still having its 5' cap. And then an poly A tail is added to the tail of this, while the intron is thrown away. This is a model of a DNA. Right here there's an enhancer that contains DNA and distal control elements. From here, activators bind to their next step. DNA binding protein comes, and then general transcription factors bind to the activators. On the activators, a mediator protein binds to there. And then at the TATA box, the RNA polymerase II binds there. And this causes the synthesis of RNA. The purpose of the enhancer is so that pretty much all of the DNA becomes stimulated to create RNA synthesis.